Juliet had a nose full of her 3 lads. Which will she choose to be her blind date? The Pheromone boy, the aftershaver, or Whitney Mr. Sweaty? The results coming up. Back to our blind date. Our lovely lady Julie is ready to choose her new boyfriend just on the basis of his smell from 3 letters chosen from a cross-section of society. Developing a sexual pheromone spray is one of the constituents and Retinol, a factory-made chemical which mimics men's natural sex hormones. And then it will slash your should drive women crazy, but does it? However, then smell of natural sweat and aftershave will lay turn her head and set. She's sick of them all, and now she has to choose her favourite.
We can take your order. So tell me about number 1. Really?
Yeah. Theo, I would say Theo.
Okay. And tell me about number 2.
He's not nice, I like that little one.
Yeah? And what about number 3?
He was between. Nice, hilarious, a bit ridiculous.
Mediocre.
Yeah.
Okay. Who have you chosen to be your blind date for this evening? Number 1, number 2, or number 3?
We've got a number 2, definitely 2.
Okay, so you chose number 2, so this means 2 that you turned down. Come in, Fabio.
Sorry.
Come in down.
Sorry.
And our next to see you is Jimmy from Scunthorpe, and he was wearing aftershave. Come in, Jimmy.
So there it is. 2 aftershave beats pheromones every time. Slash your work, boys.
Sorry. Okay. Let's see what this one is.
Hi, I'm Carmen Colombo. We're here to investigate a new product that claims to make you irresistible. So whether you're dating or just trying to spice up your love life,
They keep talking
stay
about
tuned.
the
But we're about
pheromones. And what they've tried to do, they've tried to create pheromone aftershave so that people will use it and will attract people that they shouldn't attract, or they wouldn't normally attract with their natural body odor. What they've discovered is there's about, like I said last time, there's about 150 configurations of pheromones that you can have. They've done studies with pheromones. I think I told you this last time. They've done studies with pheromones, and what they've discovered is that pheromones indicate your immune system fingerprint. So what they've discovered is that women who have a flaw in their immune system, and all people do have flaws in their immune system, they're attracted to a man that will correct that flaw, and they will find his odor more exciting, and they're more receptive to his odor than individuals who do not correct their immune system. As strange as that may seem, So potentially there's a reason for pheromones, and there's a reason why select individuals have respond to it. It's really kind of interesting. Of course, we're dealing primarily with Europeans, and of course, Europeans had some really serious problems with a lot of strange diseases, especially viral diseases. So in order to survive in Europe, where they live with their animals that create all of these strange viral infections, they had to have a stronger immune system. And of course, that may be one. It may just be Europeans or people that have been relatively directly connected with the Europeans, as horrible and strange as that may seem. But there are a lot of diseases out there. You know, the bubonic plague, we've got smallpox, you've got all these diseases that have wiped out just millions and millions of people. And of course, we do need a way to survive when these things come up, when these diseases are prevalent. Just recently, we had an outbreak of Zika virus. Before that, we had an outbreak of Ebola virus in Africa. These are all new viruses that we're deathly afraid of, mainly because they cause so much damage to people. So that is the end of chapter 8, I think, and 9. Now we're going to start with chapter 10. Chapter 10 is a tough one for me. We're talking about vision. Not that I don't understand vision. It's just it gets kind of complex when we start talking about the different structures of cells in the eye. Vision is kind of complicated, and the reason it's kind of complicated is because you've got your left eye and your right eye, and each one has two sections. You've got one that's closer to your nose and one that's closer to your ear, and sometimes they cross and sometimes they cross. But only the ones in the middle cross, the ones on the outside don't cross. As confusing as all that is. So when we are looking at something and we are creating pictures constantly, when we're doing this, everything is, for one thing, it's upside down in our eyes, and then it writes itself in our brains. Not only that, but we're taking millions and millions of pictures every nanosecond. And we are converting that into pictures, into a picture, as confusing as that is. We have 100 million rods in our left eye and 100 million rods in our right eye. We have 4 million cones in our left eye, 4 million cones in our right eye. As confusing as that is. I guess it's not that confusing, but that's 208 million pictures that you're taking constantly. You're not only looking at what is in the center of your field, but you're also seeing what's in the peripheral area of your visual field. You're taking this in at the same time. I was downstairs actually reading this chapter, and a mouse ran across my desk, but it was over here. And of course I detected the movement. I didn't exactly see what it was, and then I looked over. I couldn't tell you if it was a brown mouse, a gray mouse, or a gray mouse, until I looked directly at it, and then of course I saw it sitting there. I don't know. I don't like mice, okay? And here's this mouse sitting on my desk. Eventually, of course, when I looked directly at it, I guess my laser eyes burned it and it ran away or something. It was just sitting there cleaning itself. Until I looked away. Until I looked directly at it, and when I looked directly at it, it was my movement that made it move. I guess it thought I actually did have laser eyes. The whole area that you can see without moving your head or eyes is known as your visual field. Of course, your visual field is quite wide. Some people have really, really good peripheral vision. Mine is not too bad. I can still see both my fingers. I can't even move my finger. I can still see. My brother has tunnel vision. I told you about my brother that was a combat engineer in Vietnam, and he served 2 tours over there. He has tunnel vision. You wouldn't think somebody with tunnel vision would be a good soldier because they can't see movement. I mean, his peripheral vision is really pretty bad because his eyes are embedded deeply into his head. But he's also the one that would ambush people at night. He has really amazing night vision. So he would ambush squads of Kong or whatever, North Vietnamese, in VA. He ambushed them at night because they couldn't see him. They couldn't see very well. They didn't have tunnel vision, and he did. He had the most amazing ability to disappear at night. We used to play games, you know, we used to play Pee-wee stick down and all kinds of strange games at night, and he always won because he could hide in a shadow and you couldn't see him. It was really kind of amazing. I guess you could, I don't know, we never thought about sticking a light on this kid so that we could find him in the dark. But he was just amazing in the dark because of his tunnel vision. I have 4 brothers. I have 3 brothers, and he's the only one with tunnel vision. So the other 3 of us have normal vision. As a matter of fact, my peripheral vision is kind of amazing. But my other brother, or my second youngest brother, has tunnel vision. While the visual field may be wide, an individual's visual acuity tends to be only in the middle of the field of vision. And of course, that's what we do when we move our eyes. We're primarily moving our foveas so that we can see the center of our visual field. We concentrate on our foveas. We don't really concentrate on our peripheral vision unless you've got a mouse scurrying across your desk when you're looking at your computer screen, which is what happened to me this morning. This is because there is a concentration of cones in the middle of your visual field, and this is known as your fovea. And your fovea, of course, is trying to detect color. Color is extremely important. Color was extremely important to our anthropological ancestors because color had to do with whether food was ripe or not, whether it was edible or not. Green food, of course, is not yet ripe, usually, depending on what we're talking about. Peaches, pears, apples, of course, berries. Green berries are not edible yet. As a matter of fact, some of them are poison, and you have to wait until they ripen before they're not poison anymore. So apples, oranges, you know, all of these things. Of course, I don't know if we're talking about tropical things or not. Your fovea, when we concentrate, when we're looking at the center of our visual field, we see fairly acute vision. When we're looking at our peripheral vision, we can only do one thing at a time. We can't concentrate on everything at the same time. Usually it's fairly indistinct. So that's our visual field. However, even with the concentration of visual receptors in the center of our visual field, there is a spot where the optic nerve feeds out of the eye and blood vessels feed into the eye. Of course, these blood vessels are very small, and they're very necessary to feed all the cells in our eye. And of course, if we have a disease such as diabetes, diabetes causes our blood vessels to harden. And a lot of times what will happen with an individual, especially with type 1 diabetes, they will go blind because the blood vessels in their eyes have hardened and ruptured. And of course they lose their vision. When they first invented birth control pills were these really high doses of estrogen, and it was causing the same kinds of problems with select women. If they were pre-diabetic, they would, a lot of times they would start taking birth control pills and they'd go blind, strangely enough. Of course, they didn't give birth control pills to women who were diabetic. They couldn't take birth control pills because the estrogen was too high. Now, of course, we don't have to worry about that because we have controlled the level of estrogen. A lot of birth control pills are progesterone instead of estrogen, so we don't have the same effect they don't have the same effect that they did before. But once upon a time, back in the 60s, when they first invented birth control pills, this was a really serious problem. We had women going blind so that they wouldn't get pregnant, which seems counterintuitive, of course. The back of the eye, the area where the blood vessels feed in and the optic nerves feed out, is known as the optic disc. This is an area that maintains a blind spot in the individual's field. So when you look out, as you are looking at this picture, or you're looking at me, one of the things that's happening is that you're not actually seeing everything that you think you see. There is a blind spot in the middle of your field. Now you can make this blind spot come out by looking at select things. You can there are various things that you can do to actually see this blind spot. But the reality is our brain fills in all of the missing picture that we have. And of course it's a spot that's about this big, you know. So, but your brain is filling it in because it's got information from all around that spot, so it just fills it in. And it fills it in automatically. Normally the brain, oh yeah, that's what I just said, okay. Vision comes to humans in the form of wavelengths of light or photons. The human visual ability lies between 400 and 700 nanometers. That's ultraviolet to infrared. I looked this up before I came to class today. I wanted to find all those animals that can see better than we can. Almost all animals, depending on, well, that's not right. There are a lot of animals that can see in ultraviolet range. Deer, for example, bees, they can all see. Most birds can see in the ultraviolet range, whereas humans can't. So there are other animals that see much better than we do. Rhinoceri, for example, the rhinoceros doesn't have a very wide visual range. But then again, he doesn't really need to have one. He doesn't have to worry about predators. Nothing can kill a rhinoceros, except for humans, of course, with a high-powered rifle. So he doesn't really have enemies that he has to worry about. The other animals, of course, they can see, they have a wider range of vision because to find food and to stay away from predators. Bees can detect well beyond the ultraviolet light, and actually that's how they follow each other. A bee will go out and find a flower, and they will fly back to the hive, and they will tell all the
Snooze.
which one?
Snooze.
Thank you.
Okay.
Is yours doing the same thing? Okay. When they fly back to the hive, they actually leave a trail of ultraviolet their warmth of their body, and they can actually see where another bee has flown. And what will happen is he will come all the way back, and then the drones will all fly out, and they get the nectar from the flower. But they will follow his trail, and the trail that he has made is visible to them because they can see the ultraviolet light. There is a theory out there that birds that migrate from one place to the other, they always seem to be able to fly directly from one spot to the other spot. And we're not exactly sure why. Do they have this GPS in their brains, or what's going on? It turns out it has something to do with ultraviolet light and the ability for them to see where they're headed. And what they see is they see a spot on the horizon, and they head for that spot. If you've ever watched geese fly north or fly south, whichever the case may be. We were on a flyway when we lived in Nebraska and Iowa. It's on a flyway. And the exciting thing is that we used to see whooping cranes. Whooping crane almost went extinct, but we were able to save it somehow by not killing it, probably, for its meat. You know how stupid humans are. Stupid human tricks. Anyway, we would see whooping cranes fly over. And of course, snow geese and Canada geese and Trumpeter swans. In Iowa, we actually have nesting trumpeter swans. And they nest and produce cygnets every year, which is always exciting to my wife and I. Wandering, you know, we'll go past the pond and here's a male and a female. One morning I was driving to work and I actually saw a male trumpeter swan chasing off a Canada goose, probably the male. I imagine he was chasing off the male. But they were flying parallel to one another, and they were flying really, really fast, but really, really low. And the Canada goose was trying to maneuver to get away from the trumpeter swan, but the trumpeter swan was staying right with him. Of course, trumpeter swan's like twice as big as a Canada goose. It's huge. Well, not that big. Not quite that big. Really fascinating. A photon of light comes into your eye with select properties that we will perceive as color. The hue is the wavelength of the light. It is the wavelength of the light. Wavelengths in the 400 to 500 range are very dark purples to blue. Wavelengths in the 500 to 600 range are greens to orange with yellow in the middle. Wavelengths in the 600 to 700 range are bright reds darkening to black. Women and men have different vision. Women tend to see color better than men do. Men tend to put things together that don't really go together very well, and women will make fun of them and make really strange faces because they put really odd colors together that don't really match. I know. So why would women have better color vision? Why would women need better color vision than men? If we're looking at our anthropologic past, and we're wandering around trying to survive, why would women need better color vision than men? Men tended to be hunters. When you hunt things, you're not hunting something that's a different color. What you're primarily doing is hunting something that's the same color as camouflage. It's usually camouflage. Deer, really hard to see. Whatever you're hunting, it's probably not that easy to see because it's camouflage. But of course, camouflage is the same color. Camouflage, there is brown, it's dark, it's black, it's whatever. But it doesn't really have anything to do with color. However, the women were foragers. So they needed to determine whether something was ripe or not, whether something was edible or not. So that, of course, they needed to see the different colors. And of course, not all women were foragers and not all men were hunters. Sometimes the men foraged and sometimes the women hunted. But of course, if we're looking at hunter-gatherer groups, primarily that's what we're talking about. So the women needed better color, and that may be one of the reasons. Men tend to be colorblind a lot more than women are. Women, it's very rare for a woman to be colorblind, mainly because it's sex-linked. So if you have two X chromosomes, of course, that's a female has an X chromosome. You'd have to have the flaw on both X's in order for her to be colorblind. So it's relatively rare for women to be colorblind. But a man, of course, is an X and a Y, and his Y chromosome does not cancel out the X chromosome where the colorblindness is. So he's more likely to be colorblind. Colorblindness, as odd as this sounds, you're not absolutely colorblind or not. Usually it's gradations of, it's along a spectrum, as odd as that sounds. So there are people that can't see reds, there are people that can't see browns. Monochromats are individuals who can only see one color. Everything is shades of gray and beige, which is just bizarre to me. I had a student one time, this is when I was teaching on the Air Force Base in Mississippi. He was a monochromat. He could only see one color. This poor guy had been overseas to, let's see, where were we then? He was in the desert. I think he'd been to Iraq. Yeah, he'd been to the first Mideast War. I can't remember what it was. In '93, he was overseas in '93. And of course, they gave them all camouflage for the desert, and the camouflage for the desert was browns. And of course, we've changed all that by this time. We're far more sophisticated than we used to be. But the pattern that they used, they just used different dye. When they make your camouflage, when they made camouflage, everything was green and brown before. Green gradations of green and whatnot. So his uniforms were exactly the same, except one was brown and one was green. And his problem was he couldn't tell green from brown. You know, everything was beige and gray to him, so he couldn't tell the difference. So sometimes he'd come to work with the green camouflage top on and brown camouflage pants on. And that's why he got married, so that he had somebody who could actually tell the difference. Now the funny thing was, this guy was an electrician, and of course electricians can tell that they have to tell the difference. I know, and he worked on airplanes. That just drove me crazy. How in the world do you do this? And he said that they always put the red, let's see, there was a sequence of how they put the wires together. So from left to right rather than from, I don't know, they just bundle them together. But he had to tell the difference between red and green. And the strange thing was that he could tell the difference because there was just a slight difference that he saw. One was grayer than the other or something. It was just bizarre. Anyway, he was pretty good at his job, but sometimes he came to work with the wrong color uniforms on, especially the camouflage uniforms when he was wearing camo. This is back when your basic uniform was just olive drab, and then you had the camouflage uniforms. The BDUs, the battle dress uniform. Anyway, funny guy, funny guy. And he was blind. He couldn't tell the difference between one color and the other. It was funny. You could ask him to pick up the red pencil instead of the yellow one, and he had no idea, none whatsoever. Since most color is detected as patterns of light of different wavelengths, brightness refers to the amount of white light from the object, and of course that's what we're looking at. So as you're looking at the cover of your book, of course you're seeing the white and the red and all the browns and maroons and whatnot. Fuchsia, there's fuchsia in there. We can see all that because of the reflection we're getting off of those select areas. Everything comes off as white light, and what we're looking for is the degree of white light that is reflecting off of it. Saturation has to do with the amount of light of a selected wavelength. If all the photons reflecting from an object were 510 nanometers, the object would be highly saturated as green. 510 nanometers is green. But normally that's not the way it is. We very rarely see pure, we very rarely have pure light or pure amounts of nanometers coming off of something. So as we're looking at this, we know this is black, but of course it doesn't all look black to us because we have different reflection coming off of it. If it's in shadow, of course, we're not getting very much light coming off of it at all, and it looks completely black. But if we are looking at an area that has the white light shining off of it, it looks white. But we know it's not white because we know it's probably a solid color. Our brains do a lot of patchwork. We put things together. We assume that our brains are making value judgments constantly. So as we're looking at something, we're looking at a bag or whatever, we assume that the whole bag is the same color. And our brains are doing that to us, despite the fact that I see black, I see brown, I see gray, I see light gray, I see gray to almost maroon in your bag, but of course my brain is telling me it's all the same color. So if we could stretch it all out and get the same light bouncing off of it, we'd see exactly the same color, almost the same color. Your strap is a is a white or gray, and the other's got brown in it, I guess. So we put all these things together in our brains. And of course that makes us a really bad eyewitness, because if it's at night and we see a car drive past and there's some light coming out of a street light, which isn't very bright anymore. In the old days they were a lot brighter. We can't tell what color that car is. If it's dark, we can't tell if it's blue or green or brown or black. It just looks dark to us, and our brains are trying to tell us what color it is. It's trying to determine what color it is. The eye acts like a camera. The eye has a transparent sac called the lens that widens and narrows to focus light on the retina. Also leading to proper focusing is the cornea, or the curved outer cover of the eye that changes the light beam as it enters the eye. The shape of the lens is controlled by a set of muscles around the lens called the ciliary muscles. When the lens is made to widen or shorten to allow for focusing, it is known as accommodation. And of course, this takes place all the time. If at night you're asleep and somebody comes into your room and shines a flashlight in your eyes, of course, your eyes try to accommodate. They're trying to focus. They're trying to keep as much light out as possible. So if you do that, what will happen is the eye will turn pinpoint. The lens will turn pinpoint and you can't see. The pupil, I mean, will turn pinpoint. One of the ways that we can tell if somebody has been shooting up with heroin is that when we shine a light in their eyes, their pupils don't change shape. The reason they don't change shape is because they're already pinpointed and they can't see anyway. If we are looking at somebody and we're not exact, they're unconscious, we can't tell what their problem is. If we look at their eyes and their pupils are a different size, then we know potentially one side of the brain is doing one thing and the other side is doing something else. In other words, we may potentially be looking at a stroke. So that's one of the ways we can tell if somebody has a stroke, is having a stroke. What we'll do is we'll put them in the emergency room. We've got a select amount of white light coming down, and we will shine a flashlight in their eyes. And if they have different sized pupils, then we know that there's something probably very, very negative happening in their brains, because one side of their brain is different than the other, despite the fact that all the light coming in is pretty much the same. It also doesn't accommodate, especially the side that has had the stroke doesn't accommodate like the other side does. So we can do that. We can actually determine that. At birth, about 15% of males and 18% of females have severe enough eye problems to require correction. By the age of 20 until presbyopia, 48% of females and 32% of males require correction. That's a lot of people that can't see very well, as strange as that sounds. 50% of males and 18% of females at birth. And then by the time they're 20 years old, half of women have to wear glasses, have to have corrected vision, and then 32% of males. All my wives have worn glasses. I don't know what that means. I haven't quite figured that one out. And all three of them had really bad eyesight. Yeah, I look better to a blind lady than I do to a lady that can see real well. Nearsightedness or myopia is where the eyeball is too long and the clearest focus occurs before the light hits the retina. Farsightedness or hyperopia is where the eyeball is too short and the best focus point is past the retina. Now remember, we've got all of these people that are either farsighted or nearsighted. Okay, so once upon a time, in a land far, far away, no, it's right here. Right here, where you guys live, we're still getting the same percentages of people that didn't see very well. And this may be one of the reasons why a good hunter was sought after, because a good hunter probably had good eyesight, relatively good eyesight anyway. As I look around the room, everybody's got glasses on, but me, I'm the only one who doesn't have glasses on. Do you have to have glasses on to read or to drive? To drive and to read? You can't read without your glasses? You can't drive without your glasses? No. You can't drive without your glasses?
Right.
You can drive without your glasses? So you're nearsighted? Okay. You can see far. You can take your glasses.
These aren't even prescribed.
Oh, they're not?
I'm just kidding.
You should wear them all the time. Can you
No.
read without your glasses?
Up close, yeah. I mean,
Okay.
it's more of, just
Yeah.
to improve.
Interesting. How about you, Johnny? Can you drive without your glasses?
No, I can't drive
Okay.
that. And then I can read like close, like right here.
Yeah.
But otherwise I can't read that.
Now, this is really kind of interesting because up in Fort Belknap, which is a totally different population, but up there, almost everybody wears glasses, but they take their glasses off to read. It's just kind of strange. As you get older, of course, presbyopia, that's what presbyopia just means old eyes. Your eyes don't accommodate like they did in the past. Theoretically, and I didn't start wearing glasses until I was about 50, but I have to put them on to read, but I don't need them otherwise. I don't drive with I can't drive with my glasses on. So I have pretty good eyesight. The question is, so different populations, potentially they have different ways of, I don't know, genetically, their eyes are different. Those guys up there, they almost all wear glasses, but they have to take them off to read. So it's the farsightedness. And they're all buffalo hunters. I don't understand. I don't know where that comes in.
Yeah, it's like my dad. My dad will take his glasses off and hold a book like one of those people way out here. Yeah,
Exactly.
just to see it.
Here, can you hold this for me while I back up?
No, but there's research out there that says, it states about the food
Mhm.
they serve on reservations, like what they have in supermarkets, what they have is not very nutritious, Like,
Right.
you go to Bashas', it's like, 90% snacks and just crap, it's a bunch of processed food.
Right.
Anyway, so there's like this study that says that, like, it doesn't really, people, not just saying like, not saying poor communities, it's more relevant to the reservation because there's, what's the word for food deserts. And, you have to travel so far for food.
Right.
And, basically the brain doesn't really develop or doesn't really cognitive skills as far as, it's almost as bad as a malnourished child, so,
That's really interesting. You would expect to see the same thing in poor Hispanic communities and poor African American communities and poor white communities as well. Any place where the people can't afford really nutritious foods, you would expect to see the same thing.
Mm-hmm.
I don't know.
That could be a factor.
It could be a factor. It could very well be a factor. Of course, up there, almost everybody's on commodities.
Mm-hmm.
You guys have commodities?
Yeah.
Big commodities. Okay. A big warehouse full of food. You can go there
Still read the chapter.
and get all the cans of stewed tomatoes you want and green beans. No carrots though. Well, maybe that has something to do with it. And cheese. Best cheese in the world, I understand. I don't know. Wisconsin cheese, I guess. Anyway, I don't know. I don't understand this very well at all. And of course, if we look back, one of the things we don't want to do is we don't want to target a population as different from all the other populations, but the reality may be that, of course, it's a different genetic structure. And that's why I'm telling you about the group up north. They may have a different eyesight because they evolved hunting something different and foraging for something different. Of course, they didn't always live in Montana, I guess. I don't know. They can't, none of them can read with their glasses on. And it's weird watching them pop their glasses on. As compared to me, I have to put my glasses on in order to read. And they're making fun of me because I have to put my glasses on because they're mean people up there. They're so mean. I'm just kidding. Farsightedness or hyperopia is where the eyeball is too short and the best focus point, of course, is past the retina. And astigmatism, which my second and first wife had, astigmatism, is misshapen cornea. It's elongated, and it causes the light to pass incorrectly into the eye and cause the individual to be nearsighted. They didn't know my current wife was nearsighted until she was in the 2nd grade. She made it all the way through the 1st grade with nobody detecting it, and her name was Urity. So and of course, you know how they do. They put everybody in alphabetical order. She was sitting in the back of the room. She couldn't see anything, and she kept telling the teacher. And of course, teachers aren't real bright. They can only do things in alphabetical order. So, and they were her parents were working with her, trying to get her up to snuff because they wanted their daughter to be the most brilliant person in the world. And of course she was holding the book really close to her eyes and whatnot. And finally they took her in to see the doctor, the optometrist, and he gave her glasses, which just about destroyed her because now she has 4 eyes instead of 2. She said, oh, all of a sudden the trees had leaves. She saw the trees as just globs of green and brown before, and then she put the glasses on and she could see the individual leaves. She was so excited. The iris is a muscular covering over the cornea that gives the eye its color. The pupil is an opening in the iris that allows the light to enter the eye. The movement of the eyeball itself is controlled by the extraocular muscles, and of course they control where the eyeball goes. One of the things that's difficult to do is look directly at this. Is really stresses your extraocular muscles, and you really can't do it for a long period of time because the muscles of your eye get really tired. The darker the iris, the less light that it allows in, and the less light that it allows in, the better your eyesight. So if you have dark eyes, if you have brown eyes to black eyes, black iris, your eyesight is a little bit better in the daylight than somebody with gray eyes or blue eyes. So congratulations to those of you who have dark eyes. I have blue eyes, and of course I haven't ever had any problem. My brother that has tunnel vision has brown eyes. So, and he sees really, really well at night. He's just an amazing person. He's also got this ability to hide in shadows. And, you never even think of it. And here this kid, he disappears at night. The photoreceptor cells of most animals are part of the retina in the back of the inside of the eyeball. The retina is very thin, but still contains 5 distinct layers. And of course, this is a photograph of those layers. Of the layers. 1, 2, 3, 4. That's still 4. 1, 2, 3, 4. I don't know. I only counted 4. Not exactly sure why they don't have a 5th layer. The retina it's very thin. The first layer is the optic nerve fiber layer that is working from the inside out. So we're going from the inside of the eye to the outside, and it's the farthest inside the eye is the first layer, and that's the optic nerve layer. The second layer are the ganglion cells. The third layer are the bipolar cells. The 4th layer is the actual photoreceptor cells themselves, the rods and the cones. And then the 5th layer is made up of the pigmented epithelium. If you shine a light into a dog's eyes at night, you will get a reflection back. That is the epithelial layer. Depending on how good their eyesight is at night, will depend on what color reflects back at you. If you've ever had a dog that was old and blind, if you shone a light in that dog's eyes, then probably it came back green. And as it got blinder, the color got darker and darker and darker because the pigmented epithelium, of course, was non-functional. And since it's non-functional, the body isn't really feeding it very well. So that layer is kind of dying. The better the eyesight the animal has at night, the more likely that you will get a very bright light coming back at you, like deer, of course, have very good eyesight at night. As you're driving along the road, if you can't really see the deer because they're camouflaged and they're also in the shadows, but if they look up at you, just a little bit of light will reflect back at you. So it's almost like their eyeballs are these white or to bluish white lights. And of course, that's what you're trying to see. If you live in a place, there aren't that many deer around here, but if you lived in Montana, there's just tons of elk and deer all over the place. Montana has the second highest animal strikes per capita. So they hit deer all the time. My wife ran into a deer once upon a time. And of course, she claimed, my wife, while she's driving, sometimes she isn't thinking about driving and she isn't concentrating on the road. And she claimed that the deer was on the side and it ran into the side of her car. Right. You know, that kind of stuff happens all the time. But as I'm driving at night, of course, I'm looking for reflection because I don't want to run over a cat or a dog or whatever. If you've ever been around armadillos, of course you guys don't have armadillos out here, but armadillos, their eyes don't reflect hardly at all, and it's because they're almost blind. And of course they hunt by smell, and they're exactly the same color as the road, so you can't see them hardly at all. That's why you see so many dead armadillos if you're driving around Mississippi or Texas or Louisiana or wherever you are. Possums don't have very good reflection from their eyes either. Raccoons do, but they're yellow. They have a really good sense of smell, so they don't really need the eyesight. So this pigmented epithelium, of course, is the reflected light that you see at night. And of course that is reflecting on the rods and cones, and that is actually what they are seeing, is the light reflecting off of the epithelial layer. The rods and cones detect light and that reflects off the pigmented epithelium. That's what I just said. The rods and cones release neurotransmitters that synapse with the bipolar cells. The bipolar cells synapse with ganglion cells that form the optic nerve. And now, of course, we have that impulse going down into the optic nerve, and then, of course, it goes into the brain, and you detect it as a picture. We're not talking about one picture. We're talking about millions of pictures. Millions of little bitty pieces of a picture. That's what we're actually seeing. While most of the communication in the retina runs from outer layer to the inner layer in a vertical pattern, the amacrine and horizontal cells communicate with each other in a horizontal pattern. The reason that we have all of these different structures that are balancing what we're seeing is because your eyesight is extremely important to you. You don't want somebody shining a light in your eyes and you going blind forever. That wouldn't be good. So a lot of what's happening here is we have all of these different structures that will buffer the eyesight from whatever's happening on the outside. So we have horizontal cells, we have amacrine cells, and these function in a horizontal pattern to buffer the sight coming in, so the amount of light coming in. We have the ganglion cells, we have the bipolar cells. All of this is trying to allow us to see a proper picture. The horizontal cells connect receptor cells, the amacrine cells connect the bipolar and the ganglion cells, and all of this they can shut down if there is a problem. And we need all of these different structures in order to protect ourselves. The rods and the cones represent different functional systems. The scotopic system consists of all the rods in each eye, about 100 million cells. So we have lots and lots more rods than we have cones. And the rods only detect light. They only see white and black. So if you see a vision from your peripheral vision, you probably can't tell what color it is. There's really no reason why you could see what color it is. It's really kind of fascinating. When I'm back in Iowa, I have a treadmill, and I run, it's down in the basement, and I run on it whenever I get a wild hair, I guess. I'm getting tired. So I don't run that much. Anyway, there's a green light on the treadmill. And for the longest time, I thought it was a yellow light. And the reason I thought it was a yellow light is because I only see it when I turn off the lights. And it's down on the bottom of the treadmill. And of course, I'm trying to make my way upstairs. So I'm only detecting this light with my peripheral vision. And so for the longest time, I thought it was a yellow light. Until I looked directly at it after I turned off the light, and then there it was. It wasn't green at all, it was red actually. It's a red light and I thought it was yellow. And the reason is because I was only seeing it with my peripheral vision. And your peripheral vision doesn't see colors, it only sees light. The scotopic system is very sensitive to weak light but cannot detect color, of course. Light is captured in the scotopic system by the photopigment rhodopsin. The scotopic system is located primarily outside of the fovea, so your scotopic system is just detecting a light. The photopic system consists of all the cones in the eye, about 4 million. So if we add them all together, we've got 208 million photoreceptors in our eyes. 8 million in both eyes, or in each eye. 8 million cones and 200 million rods. This system detects color and requires relatively strong stimulation and therefore is primarily a day vision system. The photopic system is located primarily in the fovea, but is present in other areas of the retina, but in lower concentration. This makes eyewitnesses really confused because the question is, well, one of the things that we have to realize is that not all light is white. As the sun changes, as it changes positions, it actually changes the light that is shining on the earth, on your portion of the earth. So if in the summertime, at 12 o'clock noon, you have a bright white light shining down on you. But on December 22nd, when it's at its greatest angle, the light that's shining down on the earth from where we are, because we're in the northern hemisphere, is yellow. So potentially you're seeing things completely differently on December 22nd as you were on June 22nd, because June 22nd you've got white light shining down, and on December 22nd you've got yellow light coming in and shining on things. So despite the fact that there's no clouds, it's a completely clear day, you're going to have yellow light in the wintertime and you're going to have white light in the summertime. And this is one of the reasons why, as the sun is going down, of course, the light changes. The light changes from white to yellow to orange to pink, I guess, or whatever. But as the light goes down, of course, we get different concentrations of light. The photopic system is located primarily in the fovea, but is present in other areas of the retina, but in lower concentrations. And this is one of the reasons why we look directly at something if we're trying to tell what color it is. There are 3 forms of photopigments called opsins, and they detect 3 color groupings. So we have 2 photopigments. We have 2 photopigments primarily. We have rhodopsin, which tells light from dark, and we have the 3 opsins that detect color. This gets really exciting. I'm just kidding. It's not exciting at all. That's why I struggle with this chapter. What's that have to do with? Oh, it's inventory. The photopigment for the scotopic system, of course, is rhodopsin, as I just said. The photopigment for the photopic system is one of 3 configurations of opsins. All the photopigments in the eye are made up of 2 parts, retinol and opsin. Retinol is short for vitamin A aldehyde, or retinaldehyde, of course, and they shortened it to retinol just to make people crazy. Anyway, why don't they just call it A-aldehyde? Vitamin A aldehyde, I don't know. Okay, here comes the fun part. Light activates the rhodopsin molecule to rapidly lose its retinol, leaving an enzymatic receptor site in the opsin molecule, on the opsin molecule. The enzymatic opsin molecule combines rapidly with the G protein transducin. This sounds like a really slow process, but it's actually quite rapid. We see things almost immediately. But this has to take place. All this has to take place in order for us to see anything. Transducin is in turn acts on phosphodiesterase to convert cyclic guanosine monophosphate to 5-GMP. Now this sounds like a really slow process. Remember we had the system that allowed the slow system and the fast system, and this seems like one of the slower systems. Cyclic GMP inhibits sodium channel closing, and with the conversion, the sodium channel closes, of course. The loss of sodium ions depolarizes the neurons. And like I said, this seems like a really slow process, but it's taking place millions of times every nanosecond in our eyes, and of course that's why we are seeing pictures not as single pictures and then another picture and then another picture and then another picture. Or are we? Can you detect movement if you only flash your eyes at something? Do you know which direction it's going? No. I've got a Fitbit.
So does this tie in with like action potential?
Yes, it does.
But this is all within just the eye.
Well, we got 200 million rods and we got 800 million
I
cones.
hated that chapter.
So we're filling in a lot of the picture.
Mm-hmm.
A lot of it we're just filling in because we've got all these different pieces of information coming in, and yeah, we're filling in the picture. And a lot of times, if you look at something really quickly, you can't tell movement, you can't detect movement. I've got my Fitbits going out on me, and I've got some of the pixels are missing. So as what it'll do, it'll start over here and it will go all the way across. But if I'm just glancing at it, I don't see the movement. And sometimes I don't see all the pixels, of course, because some of them aren't there. But if I want to tell what time it is, see right now, I can't really tell what time it is. 10:01? Is that what you guys have? Something like that? 10:05?
10:06.
Yeah, see, I've got, all those pixels are missing, so I can't tell what time it is. My battery's dying. Anyway, so as it fills in, I have to watch it go all the way across the screen in order to tell what time it is. You can tell how many steps I've taken. Yeah. For my birthday, my wife is buying me a new Fitbit. But they won't ship it here. They won't ship the Fitbit here, so she's going to, she's having them ship it to Iowa, and then she's going to mail it to me. So, I don't know. I don't know why they won't ship it here. Anyway. Okay, so if you just glance at something, you can't tell movement. Sometimes you can't tell a lot of different things. I mean, you're just taking a photograph in your brain, and it's not a video, it's just a photograph, and you don't realize that because the brain is not used to that. The brain is used to movement. It's used to constant movement instead of just a picture. So sometimes you detect things incorrectly. Since we are dealing with millions of receptor cells at the same time, weak light will only hyperpolarize some of the receptors, and bright light will hyperpolarize most of the receptors. Thus, the response will always be graduated as to the amount of light that is detected. Now, as we're looking around the room, we've got some of our lights on, and I guess those are just this light is on. Okay, but we have, sometimes we have yellow, we have a yellow right here, and that one's all yellow. But some of these are white lights. That one's whiter than that one is. That's got a yellow thing in it. This has got a yellow. But this one's all white and that one's yellow. So the light coming off of this one, if you sat right here, you'd get pretty good light. But that one's got yellow light coming off of it. So it's not nearly as bright. There's a problem with bright white light. It makes us tired because it makes our pupil smaller. And because of that, and of course, we're trying to protect ourselves all the time, our eyes get fatigued. And that's one of the problems with your screen. As old as I am, I have my light, for my computer, I have my light turned on as bright as I can get it, because I'm old. And when somebody else tries to use my computer, it blinds them, because the light's too bright. So they always turn my computer down if somebody else uses my computer. When I was working at Ashford, we had a IT person that would try to fix my computer from time to time. And she worked in a dark office, and her screen was turned way, way down, so she wasn't really used to seeing the bright light, and then I'd bring my computer in and it would blind her. She would take her a couple seconds for her to accommodate to my bright light, and then she'd have to turn it down, of course she couldn't see. Thus, the response will always be graduated. Okay, and that is the problem. This form of response leads to sensitivity. It allows for adaptation. It allows the individual to integrate the stimulus over time. There are two types of bipolar cells that transmit information. There are off-center bipolar cells, and there are on-center bipolar cells. Okay, have you ever looked at something and then looked away and saw the after image of that thing? Of course, we played the game with the American flag. We played the game with the Canadian flag. Where was I? We played the game with the Canadian flag. And I saw the Canadian flag as purple and blue. Oh. Of course, my brain is telling, I know what color the Canadian flag is. It's white and red. Everybody knows that. It's got the maple leaf in the middle and the two red things over here, and it's white in the middle of the flag. I know what color it is. But the after image that the image that they showed me was black and green. It was black and green. So when I looked away from it, I was supposed to see red and white, and what I actually saw was a little purple, or a really dark purple, but it was still purple instead of the right color. And that's what bipolar cells and off-center bipolar cells and on-center bipolar cells do. On-center bipolar cells are activated when light is turned on. So that's what you're seeing when you're concentrating on the picture. Off-center bipolar cells are activated when the light is turned off. So when you look away, you close your eyes, and the after image that you see, that is the off-center bipolar cells. And that's what, of course, and these are relatively accurate, as interesting as that is. So what are we talking? Off-center bipolar cells are activated when light is turned off, so that the eye still has visual capabilities. It detects light coming in from the periphery of the visual field, and that is your off-center bipolar cells. They just detect light primarily, but they can detect color, and the color that they see is what the after image that you're looking at. So the on-center bipolar cells are seeing things with a lot of light, and the off-center are seeing things with hardly any light at all. Photoreceptor cells release the neurotransmitter glutamate. Glutamate is decreased in off-center bipolar cells when light is weak and turned off. Glutamate is decreased in on-center bipolar cells when light is turned on in the middle of the visual field. So we have the glutamate telling us what's going on as far as what we're seeing. And that's why we see an after image. And that's also why we can see at night. Even if we look directly at something, it is our off-center bipolar cells that are functioning and telling us what's going on. Each cone in the fovea is connected to 4 types of bipolar cells: 1 on-center midget bipolar cell, 1 off-center midget bipolar cell, several on-center diffuse bipolar cells, and several off-center diffuse bipolar cells. Each midget bipolar cell connects to just one cone in the fovea. Each diffuse bipolar cell connects to several cones. What's exciting is this. When light is turned on-center bipolar cells depolarize on-center ganglion cells. When light is turned off-center bipolar cells depolarize off-center ganglion cells. Stimulus only affects the part of the retina that is in the visual field. I know, we've got all these cells that are functioning, but remember, we have to see pictures all the time. Our eyes want to see something all the time. And we're getting 208 million indications constantly, every second. The retina will create a mosaic of the stimulation that the brain puts together as a picture. As we know, the picture comes in upside down. And the center of your right eye is actually reflecting on the left half of your brain. The right inside of your eye, near your nose, is actually reversing it, and so is the left side. So it's reflecting on the right side of your brain. The peripheral vision, the portion of the eye near your ears, isn't reversing anything at all. So this goes to your right side and this goes to your left side. However, it's your right eye that goes to the left side. So it's matching up with what this eye is seeing. I know this is really kind of bizarre. And it's flipping it upside down. So how in the world do we ever see anything? How in the world do we know what pictures look like? What pictures are coming in? Because this half is matching up with this part of the eye. And the same thing is going on over here. This part's going to my left part of my brain. This part's going to the right side of my brain. I know. So how in the world do we ever make a picture out of this strange and odd mosaic? There are various ways that the eye adjusts to different light intensities. The pupil of the eye will shrink with bright light and widen with dim light. Rods and cones have different thresholds. This is range fractionation. Photoreceptors will adjust their range of reaction depending on the intensity of the light. This will give the individual visual acuity despite the quality of the illumination. And this is one of the reasons why, whether it's December, when the light is yellow, from the sun is yellow, or it's summertime, when the light is white, we still detect the same colors. We can still look at the same thing and determine what color it is. My daughter had a light green jog bra. We went to the Grand Canyon in July. It was the 4th of July. It was 4th of July weekend. I went to visit my daughter, and we went down to the Grand Canyon, and I took pictures of her, and her jog bra, which was a real light color of green, looked almost white. The next winter, we were out at her house for Christmas, and we went up to Camelback. She lived in Tempe, and we went to Camelback. She used to run up Camelback, see how fast she could get to the top. I don't know. She was only like 22 years old, or 25 years old. She just graduated from college. Anyway, and I took a picture of her then. And her light green jog bra looked darker green. It was a darker colored green. It's really kind of interesting. Why? Because it was a yellow light that was reflecting off of a light green top. And since it was yellow and light green, it came out as a much darker green. The jog bra, exactly the same jog bra, strangely enough, that I took a picture of in July, looked almost white because it was a white light coming down, reflecting. It was almost noon in both cases. And one of them looked like a darker green, looked like a different bra. But the reality was that it was exactly the same jog bra that she had on. Can I say that? Sorry. I took a picture of my daughter in her bra. That didn't sound quite right, but it's a jog bra. And, she's an athlete, and she doesn't usually wear a top. She does wear a top. She wears the jog bra. But she exercises in her jog bra. Okay. Anyway, really fascinating stuff. Calcium is a major player in photoreceptor light adaptation. When an individual changes from one extreme light to another, it takes time to adapt to the new intensity of light. This is because, everybody's leaving already. This is because the retinol and opsin break down at a higher rate in bright light and must regenerate to be available in the dark. And this is why dark adaptation takes so much longer, because you don't have as much retinol and opsin. But the possibility is, if you go out in the dark, your eyes will adapt. If you wake up in the middle of the night, it's dark in your room unless you've got all kinds of lights on, and your eyes will adapt. When you first go to bed, you're blinded because you've just turned off the light, so you don't have enough retinol and opsin in order to see. So it's almost black.
So this can also tie into the circadian rhythm? Is that how your body can tell
Exactly.
light of day, or when to go to sleep?
Exactly. So if we have artificial light on all the time, it kicks us off.
Yeah.
By about a half an hour a day.
Really?
Yeah. Well, I told you about being in that cave, that damn cave. And the light wasn't white, it was yellow. It was a yellow light. Back then, we didn't really have these lights. So the light was yellow, and it really changed things. Made people look different, like, well, their skin looked different. So everybody looked jaundiced because the light was yellow. I know, it made everybody look sick. It was really kind of strange.
Vitamin D too as well, right? Is that what you get from sun?
Oh man, lucky me! We weren't in the same cave. My wife was stationed at Zweibrücken, and I was stationed at Ramsey. So we weren't in the same caves. We were in the same area, but we weren't in the same caves. It was really kind of strange. Well, I thought that the caves were connected, so I'm trying to find my wife. I'm going all the way back to the back and trying to find my wife. It just went down. It's kind of like Fellowship of the Ring, where they go down into the caves. It was so bizarre. I never could find her. And they were afraid I was going to get lost. Adaptation is much quicker when adjusting to dark to light because the retinol and opsin level is not depleted. In the dark, of course, it doesn't deplete. So if you go from a darkened theater to outside, you can see fairly well. But if the opposite is happening, if you're going from the outside into a theater, you can't see, you're bumping into people, spilling your popcorn, all that horrible stuff is taking place. You see where I am. Okay, why don't we stop right here? We'll pick this up next time. Sorry about this chapter. It's really confusing. There's a lot of different things that are taking place. And the reason all those things are taking place is so that your eyes will adapt to seeing and